Thanks, Johannes, for the kind introduction. So today I'll be sharing some of our recent work on modeling the 3D genome. So my lab works on the problem of understanding genomic sequence using computational approaches, especially deep learning approaches. So we are at the time that we know not just the genomic sequence, but also have an abundant amount of data that measures different aspects of genomic properties and biological processes. But for most of the cases, our understanding about how those processes depend on the genomic sequence is still rather incomplete. So it is a great opportunity to develop computational models that can capture the dependencies from sequence to those various biological processes. And today I'll be focusing on our recent work on understanding the sequence basis of 3D genome architecture. So we now know that our genomic DNA is highly spatially organized in different spatial scales. And a lot of it is learned from assays such as Hi-C, which allow us to measure the 3D genome by sequencing approaches. So essentially, we can fix the genome and cut them into fragments and then ligate the genomic fragments that are in close proximity to each other. So then we can sequence to detect those close contacts between genomic sequence fragments. So then by counting the frequency of those contacts, we can obtain those heat maps like this. So here, both x-axis and y-axis represent different genomic locations. And the diagonal will represent just interactions with itself, and the all diagonal positions represent interaction strengths between two genomic locations. So by visualizing the Hi-C data in this way, we can see those three characteristics of 3D genome interactions in different spatial scales. At the most local scale, we see structures such as those individual loop interactions. And we zoom out a little bit, we can see those topologically associated domain structures. So where in each individual domain or TAD, you see interactions preferentially happening within the domain, but not between domains. And if we zoom out even more and look at scales more than 1 megabase pair, the characteristics of the 3D genome interaction change again. So now the main driving feature that we see are these plate-like or checkerboard-like patterns. So we can interpret this as having two compartments in the genome, and the sequencing one compartment preferentially interact with the same compartment in different parts of the genome, but not with the other compartment. So we can divide the genome into the A compartment, which is more enriched in active genes, and the B compartment, which is enriched in inactive and or depleted of genes. So like everything in biology, we know that the structure is highly relevant to the genomic function. And we know that the 3D genome is involved in transcriptional regulation, in DNA replication, and DNA repair. So we also want to understand how the 3D genome is dependent on the genomic sequence. So we know that there should be a strong dependency because 3D genome structure, like many other genomic properties, are highly genomic location specific. So there has to be a reason that drives the specificity of a 3D genome at different genomic locations. And that is very likely to be the genomic sequence. So you want to understand the sequence determinant and be able to utilize that for different genomic sequence understanding and understanding the 3D genome. So we've formulated the question as a machine learning problem. So simply, we want to take genomic sequence as input and predict the 3D genome structure at different spatial scales. So previously, there have been excellent works that have enabled the prediction of the 3D genome structure at below 1 megabase scale, but it has still been a challenge to be able to predict the 3D genome structure at all the different scales. So that is a challenge that we resolve with this new development. And of course, this is a very challenging problem because that we have to deal with very large input that we want to be able to predict the 3D genome structure from up to entire chromosome. So it has very large input. It's almost probably the largest input that has been given to a deep learning model that I've known. And at the same time, that we also have 3D genome data that can also be very large. You want to be able to predict the structure for the entire genome together. So we have to deal with those computational challenges. And we approach this challenge by designing a multi-scale prediction mechanism that is kind of like Google Map. So we know that at the longer range, so those genomic interactions are measured at lower resolution. So we don't have to represent them at the finest resolution. So we can predict the 3D genome interaction patterns for a large genomic location regions, for a large genomic region with relatively low resolution. And then we iteratively zoom into subregions, smaller and smaller subregions, and predict the 3D genome patterns with higher and higher resolution. So in this way, at every level, we predict a different size of genomic region at a different resolution, but the model will also be handling the same amount of data in prediction. So in this way, we can generate predictions kind of like a zooming process. We start from a large genomic region and iteratively zoom into the genomic location of interest. So to pair with this zooming series of predictions, our deep learning architecture utilizes a hierarchy of sequence encodings generated by convolutional neural networks. So in this hierarchy of sequence encodings, at different levels, sequence encoding is computed from a different size of sequence input. For example, at the lowest level, one value here is computed from a sequence of 4 kilobase pair, whereas at the top level, one value can be computed from up to 1 megabase pair sequence. So we use the sequence encoding at the corresponding level to predict the 3D genome structure at the corresponding resolution. And then we iteratively generate finer and finer resolution predictions, utilizing input from the sequence encoding and also predictions from the higher level so that we can make sure that the prediction can receive information from the entire large sequence input. So in this way, we can evaluate, we can train this model, and now we can evaluate its performance by giving it a chromosome that we have never used during training. So this is an example of such prediction, starting from the 32 megabase per level. And this region is a 32 megabase per region. And each plot on the left is predictions that is coming from a subregion of this 32 megabase per region. And we iteratively zoom into the center of those regions. And at the finest scale predictions, we can see clearly this pad structure. And while we zoom out, at larger spatial scales, the computing compartments structure becomes the dominant features. So we can predict those, we can now predict those 3D genome structures at all of those different spatial scales. And if we compare with the actual experimental data measured on this chromosome, we see that actually there is very good agreement with our prediction. So we can now actually do very good predictions just using only sequence as an input across all of those spatial scales. And here, just to show you the predictions at even larger scales, up to the entire chromosome, like the whole chromosome 9 is 138 megabase pair, and we can predict up to 200 megabase pair scale, which will be able to allow us to contain the even largest human chromosome. And also that we are also allowing the prediction of interchromosomal predictions at those larger scales. So if we evaluate the prediction performance quantitatively by comparing all of our predictions with experiment and compute the pyramid Pearson correlation, we see a rather consistent correlation across different spatial scales. And we also observe that in addition to being able to predict the most common form of 3D genome interactions, such as those CDCF-mediated interactions and TET domains, we also find that the model can capture some of the rarer forms of genome interactions. For example, in this case, we see that those interactions are actually polycomb-mediated interactions, which are overlapping with the H3K27 trimethylation. And these interactions are also cell type specific. So you see it in H1 embryonic stem cell, but not in the other skin fibroblast cell type. So when we train the two models, one for the embryonic stem cell and one for the skin fibroblast, we also find that it not only predicts the polycomb-mediated interactions correctly, but also captures the cell type specificity so that it predicts these interactions in the H1ESC, but not in the skin fibroblast. So this is an example, another example of enhancer-promoter interactions, where we can see these interactions overlapping with the enhancer and promoter histone marks. And in this case, we can also generate the cell type specific predictions of those enhancer promoter interactions, which is now observed in only the skin fibroblast cell type, but not in the H1 embryonic stem cell. So now we can predict the 3D genome interactions at almost all the scales that can be measured through high-C type approaches, and also capturing all those different types of interactions. But what is the use of the model if we have already measured all of those interactions already, right? So the main motivation of training such models are, I think, twofold. So, but all of them, both of them are based on the model's capability of being able to predict from new sequences, genomic sequence that we have not used in the training of even sequences that we have not seen before. So for any of the sequence as input, in principle, the model can predict its 3D genome structure at different scales. So this allows to both use it to predict the effect of genomic variant, including large structural variant, and predict its impact on the 3D genome, and also use it as an approach to generate hypotheses about the sequence-based mechanism of the 3D genome organization. So we know that the larger structural variants can lead to different diseases, including here in this example, many limb malformation phenotypes through its effect in 3D genome. So to validate how our model can recapitulate the 3D genome interaction effects by those structural variants. So we collected all of those prior studies that has studied the known effect of those structural variants and compare it with our prediction based on the sequence. So this is one of my favorite examples from Stefan Modulo's lab, where they studied the structural variants in this region, which leads to very different phenotypes. So here we will try to use the 3D genome model to be able to recapitulate and explain the phenotypes that's generated by these 3 different types of structural variants. And here is the region, and the 2 genes that are involved here are the KCNG2 and the SOX9. And here is the prediction versus the experimental measurement of the 3D genome structure in this case. We can see that these 2 genes sit in 2 different TAD domains. So now, first predicts the effect of the first type of structural variants that leads to a sex reversal phenotype. So if you duplicate this region, so if you duplicate either up to this longer range or minimally duplicate this region, it leads to a sex reversal phenotype. So we can predict the effect of this duplication by introducing it to the genomic sequence. So we can just generate a synthetic sequence that contains this duplication and ask the model to make predictions for this sequence and compare with predictions from the wild type sequence. And as we see here, the duplications of both the long form and the short form is enlarging this TAD, right? And the genomic interactions within the duplicated regions is duplicated. And especially the genome interactions for this putative enhancer that sits in the minimal duplication variant region is duplicated, and the interactions with the SOX9 is expected to be increasing because of this duplication. And this is also consistent with experimental measurement. So here it is showing an experiment done in mouse that introduced a homologous equivalent duplications. And as you can see in the high C measurement here, that the interactions within the duplicated region increase. And not only that, the interactions within the same TAD domain also increased, which suggests that this duplicated region is located still within the same TAD. So different from what you can see here, where we resolve the 2 different copies of the duplication, by short-read sequencing-based high C measurement, you cannot distinguish between the 2 duplicated copies. So therefore, it is slightly harder to interpret, but it still is consistent with the story here that it leads to an enlargement of this TAD domain. So we now go to another interesting variant where if we duplicate an even larger genomic region, which contains the region where duplication leads to sex reversal, this leads to no phenotype. So even though this duplication is larger and contains the sex reversal duplication, it leads to no phenotype. And this can be clearly seen in this. The reason can be seen from this prediction, where the duplication has moved the SOX9 location to here. And it is still interacting with its original interacting partners because of the introduction of a new TAD boundary here. So SOX9 was initially interacting with this part of the genome, and here the interaction patterns of SOX9 is actually preserved. So different from the sex reversal duplications where the SOX9 TAD domain is enlarged and gain new interaction. Here, the original interactions is preserved. So therefore, it leads to no phenotype. And this is also consistent with the experimental measurement. And if we move beyond this sex, those no phenotype duplication, and if the duplications is a bit longer and reach past the KCNG2 gene, yet another phenotype appears. So it will lead to the Cook syndrome, a finger malformation phenotype in this case. So if we have an even longer duplication, which leads to the duplication of KCNJ2, now we have 2 copies of KCNJ2. And even though SOX9 is still unaffected because of the same reason, the KCNJ2 is now, the new copy of it is now moved into a new tag domain, which interact with some of the prior partners of SOX9. So it can actually be experimentally shown that the KCNJ2 has now hijacked the expression pattern of SOX9 and also show gene expression pattern that is similar to SOX9. So in those cases, we see that the we can use the 3D genome predictions to help recapitulate the known effects of those structural variants on the 3D genome. And here is just another example of a complex region where several different structural variants all leads to different phenotype, and our model can predict that in those different cases, these different structural variants actually leads to, they all lead to interactions between another gene with this putative enhancer region. So in different structural variant cases, they moved a different gene into interacting with this enhancer, and therefore explaining its different phenotype. And it's also consistent with the experimental measurements through the 4C-seq, which measures the 3D genome interaction patterns from a single viewpoint. And we compared side by side the 4C-seq experiment and with the high-C, with the predictions from our model. You'll see that it recapitulates the experimental effect of those structural variants. So there are also more examples. I won't show more of them, but we have really tried to do it with all of the structural variant examples that I can collect. And in almost all of the cases, we see that there is a strong consistency between the sequence-based prediction and the 3D genome. So I cannot say this for all the sequence models that I've developed, but in this case, we think that it's actually really closely approaching the measurement that is that can be done through experimental approaches. So I think it can be a very useful tool for studying the effect of structural variants in 3D genome, even prior to obtaining the experimental data. So let's move beyond the application of predicting the effect of structural variants on the 3D genome. And obviously, another question that we will be very interested in is that now the model can very accurately predict the 3D genome structure from the sequence. And what is it using to generate such prediction, right? And can we learn anything from it and to generate hypotheses about the 3D genome prediction? So how do we do that? I tend to think of the 3D genome interactions as a studying the 3D genome interactions through the computational model as we can consider the model as kind of a virtual cell. So we can do virtual genetic screens on the model. So we can design all kinds of mutations that we want to screen, and we can introduce them into genomic sequences and generate those synthetic sequence. And then we can use our computational models to predict the 3D genome structure carrying those mutations, right? And then we can analyze our screen if result. And because we have a computational platform for this, we can do this very quickly. We can also quickly iterate and design new virtual genetic screens. So to improve our understanding to how the model is generating predictions based on those sequences, right? So we did this and use it to study the sequence determinants of the 3D genome at different spatial scales. At different spatial scales, different screen strategies has to be designed. So first, at the more local scale, we design a strategy that mutate every 10 base pairs in this region, not just in this region, but also across the entire genome. So we mutated all 10 base pairs in the genome, and actually every 10 base pair is mutated 3 times, and we take so we randomly shuffle the 10 base pairs 3 times and takes the one that with the minimal effect. And this gives us a track like this, which show us the effect of that 10 base pair sequence on the 3D genome structure at every genomic location. So first of all, first we want to compare with what is known to be impacting the 3D genome, the CTCF binding sites. And we can see those strongest sites are all collaborating, all overlapping with the CTCF binding site. And if we analyze quantitatively, we also see that if we look at those tiers, highest tiers of locations with strong genomic 3D genome impact, these locations all have a strong CTCF motif within or nearby these 10 base pair sites. But if we look beyond the strongest sites that has effect on 3D genome and look into those moderately impacting locations, we see there are also a lot of sequences that are not having a CTCF motif nearby. And when we look at the motif enrichment in those regions, we find that there's actually a very cell type specific enrichment. So when we look at the enrichment of motifs in H1 embryonic stem cells that impact the 3D genome, we find strongest enrichment of the POV5F1 SOX2 motif, which is known to have a strong important function in embryonic stem cell, whereas in the skin fibroblasts, we find that the strongest enrichment was from the AP1 family motifs. So which is also consistent with the known biology for the skin fibroblast. And we look at those motifs in the same way, we see those cell type specific enrichment of those POV5F1 SOX2 motif and the AP1 motifs in the corresponding cell type. So those are the hypotheses that we generate from the model, and those are still, I think, questions that I think remains to be further tested through the experimental approaches. And on the other aspects, if we look at the larger scale, it is largely, I think, still a mystery of what are the sequence determinant of the chromatin compartment. So even though it is known and expected that the gene expression has a strong relationship with the chromatin compartment, because compartment A is enriched in active genes, and activating genes can lead to switch of the genome compartment. But it has been hard to test at the sequence level what are the sequences we remove will lead to a chromatin compartment change. But we can test that in silico through these computational approaches. So in this case, we also train a model that is a special data set that is depleted of TAD by cohesing depletion. And that data set contains only chromatin compartment structures, which is largely unaffected by the cohesing depletion, but the TAD structure is totally removed so that we can now deconvolve the impact on the TAD level and impact on the chromatin compartment level. And utilizing this model, we design an insertion screen where we insert segments of genomic sequence into different locations in the genome and see whether this insertion leads to a chromatin compartment change, which can be detected through the 3D genome structure prediction at higher scales like 32 megabase per scale. So I'll show you the example of screen in this region where all the source sequences are about 10 kb segments coming from this region, and we insert all of them into 9 different locations that covers both A compartment and B compartment in this location. And we can visualize the effect of this insertions in this way, where for each row is corresponding to a single target location, and the x-axis represents the locations of the source sequences, and the y-axis represents its effect on the 3D genome. So we can think of it as a chromatin compartment activity profile, and we definitely see that this profile is having two very different patterns depending on the chromatin compartment location of this target location. So if we look at the targets that are coming from the B compartment, it is sensitive to B to A compartment transition. So therefore, it is mostly detecting sequences with compartment A activity. Whereas if we look at the target location that is located in the compartment A, it is mostly sensitive to compartment A to B transition. So it's mostly detecting compartment sequence with compartment B activity. So we see that largely the compartment A activity sequences coming more from the compartment A, compartment B activity sequences is coming more from compartment B, but it's also more widespread. And at the same time, the sequence with compartment A activity is much sparser compared to the sequence with compartment B activity, right? So now we want to look deeper into what leads to the difference in the characteristics of the compartment A and B activity. So first of all, we want to know what are the minimal insertion size that can lead to such chromatin compartment activity. So what is the size that is sufficient to change the chromatin compartment, in other words? So we screen for different insertion sizes, and strikingly, the chromatin compartment A activity requires relatively short sequence. Even up to 400 base pairs still preserve a substantial proportion of this activity, whereas for compartment B, activity really requires pretty long sequences that is at least about 10 kb long, and it decreases very quickly with the shorter sequence insertions. And then what is the sequence here? Like, what is the location of those sequences with compartment activity, right? So we see that it's actually coinciding with the transcription start site activity very well. So it has a very good agreement with the cage measure transcription start site activity. And if we zoom in into these locations with the strong compartment activity, the actually exactly center at the transcription start site. And we see a several orders of magnitude difference between the locations with strong compartment activity versus those that without compartment activity. So that it has a very strong agreement with the transcription start site locations. And also we see generally no dependency on the direction of the transcription start site at any target sites. And at any target site, the transcription from both positive strand and negative strand has the same level of effect on the compartment according to the model. And lastly, we also want to detect what is the sequence pattern size. For example, whether motifs are involved or larger patterns, larger than regular motifs is involved in the compartment activity. To do this, we use a permutation approach. So we still insert the 10 kb sequence, but we will permute the sequence by blocks of different sizes. So the idea is that if we permute the sequence by a certain block size, we destroyed all the sequence patterns that is beyond the size of the block. So we permute the sequence by 100 base pair blocks. If a sequence pattern of 200 base pair size is required, then that pattern has to be destroyed by 100 base pair sequence permutation. So therefore, if the sequence activity is preserved at a certain block size, it means that the sequence does not depend on the activity does not depend on sequence patterns that is beyond that block size. So in the compartment activity A, so the compartment A activity, what we see is that the compartment A activity strongly decrease after permutation that is below 100 base pairs. So it means that it needs sequence patterns that is at least 100 base pair and probably more optimally beyond 200 base pairs to maintain the compartment A activity. Whereas for the compartment B activity, it is actually quite robust to permutation at different sizes. So even if we go down to permutation of segment of 2 base pairs, it still preserves the compartment B activity. So it seems like the compartment B activity is not very dependent on any complex sequence patterns, but rather it has a strong consistency with the percentage of ATs within each of the 10 kb regions. So with higher AT percentage, you'll have a higher compartment B activity. This is also something that is previously known. So lastly, we see that if we permute long chunk of sequences in different genomic regions, so we permute 1 megabase pair sequences, just shuffle to remove all of the sequence patterns, we see that these permutations at the compartment A locations lead to B compartment formation, here and here, whereas permutation at the compartment B locations leads to no effect. So which kind of leads us to a overall hypothesis about the sequence basis of compartment formation, whereas compartment A is sort of the active compartment that is driven by the sequence from the transcription start site. So even a short sequence at the transcription start site likely leads to some widespread effect on the chromatin compartment, probably because of the transcription start site also leads to a more widespread chromatin level change. And in contrast, the compartment B seems to be the passive side of the compartment, whereas it does not need to have complex sequence patterns and beyond just AT content. So it requires sequences that is at least about 10 kb long that does not have the compartment activity to form. So I think one remark I want to make here is that it's interestingly also consistent with the size of about 10 kb is also consistent with experimental approaches that measures what is the minimal size of genomic sequence fragments to maintain a stable chromatin compartment. So we'll fragment, generate genome, we'll chop the genome into fragment that is beyond 10 kb, then the chromatin compartment structure is largely preserved. Whereas if you cut the genome into fragments that's shorter than 10 kb, then the chromatin compartments start to dissolve. So that's the result from Job Decker's lab. And this length scale is interestingly consistent with the length scale the model predict to have a strong impact on the chromatin compartment, and maybe is related to the length scale that is necessary for the mechanism of compartment B formation. So we think that the sequence models of multiscale 3D genome is a useful tool for the study of the 3D genome. And we have talked about various aspects of applications using the 3D genome sequence model, and also make it available through a web server. So you can actually just submit any structural variants that you're interested in. Currently only support simple variants. If you are interested in more complex variants, you can also do it using the code that is provided in our GitHub repo. So I hope that if it's relevant to our research, that you can make use of it and see whether it is helpful in helping understanding the effect of those variants on the 3D genome structure. So lastly, I also have a little advertisement for another recent tool that we developed for mapping the sequence regulatory activity. So in this case, we developed a sequence model, and that is aimed to be able to tell the regulatory activity for any type of genomic sequence. We grouped the activity into 40 different sequence classes, and you can predict whether a variant is impacting, for example, enhancer and promoter activities through this approach. It's also our most recent and best models in telling the regulatory activity of the sequences, which is also having a web server and code available. And I'm happy to help if you are interested in utilizing those tools. So lastly, I would like to thank my funding sources, and most of my lab members joined after I've finished a large part of this this work. That's pretty much done by myself, and the last part of the regulatory activity model was done in collaboration with Kathleen Chen in Princeton University. So I'm happy to take any questions.
Thank you very much for this excellent presentation. This impressive work, and I can see this virtual screen to generate many hypotheses for other biological systems. This presentation is now open for discussion. Please either raise your hand and I will allow to unmute you and you can ask your question yourself, or you can type your question in the Q&As or in the chat and I will ask it for you. I will start the discussion with one question. It's fascinating how detailed your resolution is. How many differences do you observe between prediction and observation? Are there certain areas where your in silico method has problems to predict the interaction? What do you need to improve to improve the performance in these areas?
Right, so that is a great question. So we also try to see what are the cases where the prediction get it wrong. So at the more local level, like TED level, it is actually fairly infrequent that the model get it wrong. The places that we find that has a strong difference between the observation and the prediction actually tend to be cases where there is a mapping error, like where there is a highly repetitive region. So there is some high-C mapping error in that area. And the predictions for I think the prediction for the more special type of interactions, and mostly the enhancer promoter interactions still have space for improvement. Like here, I think the predictions largely capture the characteristics of the enhancer promoter interactions, but not exactly ranking the size of different interactions in the correct order. So I think there is still space for improvement on this aspect. And also, if we look at the larger scale predictions at the compartment scale, so there are still some places where the prediction is not perfect. Like here, that it has some difference with the observation. So I definitely think that there is still space for improvement is across all scales, but especially at the higher chromatin compartment scale.
I have a question from the Q&A from Ashish Jain. Can you talk more about the sequence encoding technique, like the sequence length and the padding and so forth, and that you used for this model? And did you find any issues while encoding long genomic sequences?
Right, so that is a great question that I didn't go very detailed into the technical aspects. So the sequence encoding is using, so we use very long genomic sequences, and the representation is relatively simple still. So even for 32 megabase per sequence, for example, encoded by a 4 by 32 megabase per array, where we use 0 and 1s to indicate the sequence, whether it is A or T. And the challenge in doing this is, as I mentioned, the size of the input. So actually, if you use regular deep learning approach, you cannot even compute a single layer of computation because the GPU does not have that much memory in it. So to be able to allow this, we actually utilize a special type of technique for deep learning training. So for deep learning training, using a small amount of, that's requiring a much smaller amount of memory. So instead of doing the traditional layer by layer computation, we utilize a block by block computation. So each horizontal block can compute a block for the several layers using only a small fraction of the entire sequence, so that we don't have to process the entire 32 megabase per sequence at the same time. For example, we can compute half a megabase per at a vertical block, and then we move the execution horizontally to make computation for the entire network. So it still preserves the same computation as original, but now require much less memory. So this technique, which we call horizontal checkpointing, is key for being able to train and use the sequence model at this scale.
Great. Wangqing Xiao.
Oh, hi. This is Wanqing from Research Computing. Very interesting talk. Thank you so much. So I have like two questions. The first is about the cell type or tissue specific prediction. So it is really nice to see that your algorithm could do that. But I was just wondering, like, would it be possible, instead of using Hi-C from different cell types, combine Hi-C with maybe epigenetics data or like transcriptomic data, which are kind of like more widely available at like maybe cell type of interest, and then perform the prediction and get the like kind of like more context specific result?
Right. So that's a great question. So for models that's like ours, that has to be trained with Hi-C data from that cell type. And for other cell types, if we have epigenetic data available, like whether we can use those kind of data as the input to predict Hi-C, I think it's definitely possible given the strong correlation between Hi-C and the epigenomic marks. And one thing to note is just that the models that are trained from not just sequence, but also other type of input, for example, epigenomic marks, can having some issues in capturing the correct causality direction. So for sequence-based models, there is one key feature that is needed for us to make prediction of structural variants, that is capturing the right causality direction. Right. So if we want to predict structural variant consequences, then we need to make sure the predicted effect is causal. Mm-hmm. And the sequence is generally, we consider it as the upstream of all of the different regulatory phenomena and biological process. It's not the consequence of the biological process. So that the sequence-based models, as we expect and generally see in our test, is that it's correctly capturing the causality direction, whereas other genomic measurement, including the epigenomic measurements, it can be both causing and be a consequence of 3D genome structure, right? So if we predict the 3D genome structure from the epigenomic marks, we are not certain that whether the prediction is using the causal direction or the other way around. So that is okay if the goal is to just getting the 3D genome prediction for that cell type But if the goal is to predict the effect of structural variant and mutations, then it has to be taken with greater care.
I see. Yeah, that makes perfect sense. Yeah. So my second question is more about the application. So potentially we might be interested in using this to kind of like do a high throughput screening for like maybe thousands of patients' data. So in that case, what would be your kind of like recommendations for the kind of like analysis design for that? Shall we kind of like start with the structural impact score, identify the key regions, and then go through the viral inspection, something like that?
So to screen for structural variant effect, start seeing like screen structural variant effect across a large set of variants across many patients, right?
Yeah, and potentially,
that is the application.
Yeah, and potentially maybe like variants smaller than structural variation, if that's also possible.
Right. So you can summarize the 3D genome effect through the structural impact score. And in addition, that I think it is also helpful to consider the locations of genes to only look at the structural changes at the location of actual genes. Right? So you don't have to look at the structural change at all locations, but more focusing on, instead of just ranking the individual structural variant, you can rank by structural variant and gene pairs. I think that could be a good approach to screen for those large structural variants.
Cool, I see. Yeah, thank you so much.
Yeah, and I'm also happy to discuss more if you are using the tool for larger scale study. So.
We currently don't have any other questions in the chat. I think there was a beautiful discussion, great presentation, great work. Thank you so much for being with us today. And
thank you for the invitation and all the discussion. It's wonderful.
Yeah, and thank you for presenting this work and giving more insights into this topic. We will then close the session today. Thank you so much for attending, everyone.
Thank you, guys.